Hello, and welcome to our lecture on RNA transcription in our microbial genetics lecture series. And now, midly, RNA transcription is where we start to deviate from DNA replication and, uh, our pulling apart analogy that makes much more sense. And now we have to start thinking locally. Remember how we talked about the phenotypes and how you can have an eye cell and a liver cell from the same DNA based on what is actually getting expressed. What do we mean by that? We mean that it's actually going to pull apart at one little spot that encodes for a protein. Okay, it can be 2,000 base pairs, it can be 5,000, 10,000. It can be a lot of base pairs to make a big full-size protein that is going to catalyze some reactions inside of a cell. Okay, but it kind of will make sense still, I promise, if you think about it in three distinct steps of replication. And what will I get if I just need to replicate this DNA and I'm pulling apart? What am I going to get with transcription, and what's going to be my end result? It's going to be mRNA for the hint. Okay, and then when I take that mRNA, what's going to be my final result after translation, right? It's a protein. Okay, so again, that leads to our once again review on the central dogma, okay, where we need to go from DNA, make RNA. That RNA needs to get processed, cutting out introns. We don't need those. Okay, those introns get cut out, and as they get cut out, we leave the nucleus if we're a eukaryote, and if we're a microbe, we're going to start right away. Right? We're not going to even wait for introns to get processed. We're going to just get started right away. But either way, we still went from DNA to RNA. And what do we get started right away on is translation. Okay? That translation will occur once we are out of the nucleus if we're a eukaryote, because the ribosomes will find this, and it also happens in prokaryotes just right away. Okay? What do I mean by that? Well, look at this picture here. As it is just being transcribed, transcribed, that is being transcribed, the ribosomes find the strand and make the protein, and make the protein, and make the protein, and make the protein. Okay, that's what they do. But in the eukaryote, it takes it to leave the DNA, the nucleus first. Then, when it leaves, it can be processed by multiple ribosomes at a time. Okay? So prokaryotic transcription and translation occur simultaneously as it is happening. That is not the case for eukaryotes, because eukaryotes have to get it out of the nucleus. Okay? And we have many ways to control this type of gene expression. Again, what do I mean? I mean that we're only unfolding here, and we're only unfolding here. Okay, why didn't we unfold here? With the exception of it being junk DNA, and there's an actual gene there, we're not turning that on because it's not going to help us any. Okay, so we don't need it now. Alright, so now we're going to get into the diagram of right there where I keep pointing to, right where it all starts. What do we need? We need somewhere to land. Just like our DNA polymerase needed a place to land, our RNA polymerase needs a place to land. Okay? We call that usually the Tata box. Why do we call it Tata box? Because I just underlined it. It looks like Tata, TATTAA, that is the TATA box, TATTAA. And then you can also add a T if you'd like. All right, that will be typically 10 base pairs behind the transcription start site. Why 10 base pairs? Because that's how big the surrounding protein is. To land on it and to make a site that it can land on and be nice and succinct. Okay, that is called initiation. We are finding the TATA box. And now that we found the TATA box, we will start to transcribe. This is also called RNA elongation instead of DNA elongation. The RNA is going to elongate, okay, and that is where we get the name polymerase from. It is going to create this polymer, the RNA polymer. It's going to make a polymerase. It's going to go in our same 5' to 3' fashion, okay, and it will copy the strand as it goes on. It's going to copy it, and instead of T's, it will use U's. Okay. As we said, in eukaryotes, and only eukaryotes, does the RNA get processed. In prokaryotes, RNA translation can happen, or translation from an mRNA strand, from an RNA strand, can happen right away. Conversely, in eukaryotes with an mRNA, it does not happen simultaneously. It needs to get out of the cell. It needs to be told where to go, and it uses these features, untranslated regions, 5' cap, and poly A tail, to tell it where to go. Okay, and we also cut out these things called introns. Introns are not needed. They're just in the sequence, but they can be removed. All right, and so that is RNA transcription. Okay, remember, we needed an RNA polymerase to make an RNA transcript. We needed a DNA polymerase to make a DNA replicated strand, right? Okay, so RNA polymerase makes RNA. RNA, as we'll see, will make protein. Okay, and I will see you in the next lectures.